Okay, so I guess I'm going to start now. So this is a talk that was it's combined. It's two talks that are kind of combined into one. And I generally give sort of long talks to begin with. And so I tried to cut out a bunch of slides, but I didn't completely cut them out. So within the PowerPoint online will be all the talks, all the slides from the two talks combined. The Basically, there's a lot of extra slides that are hidden that you can access if you download the talk online. But at least I'll just show you the ones that are not hidden, of course. Anyway, I'll get through as much as I can. I promise I won't take more than an hour. And also, please stop me if you have any questions whatsoever about anything. And I'll try to be as clear as possible. And a lot of this, so looking at the audience, a lot of this you already know. I'll try to add new insight or maybe some new cutting edge sort of applications or things like that from my own perspective. And also just to reemphasize, so what I do is pretty much contrast mechanism work. I push the limits of the technique. I try to think about how the acquisition and the processing can push the limits of resolution, both temporal and spatial resolution. I like to develop interesting paradigms that sort of help pull things out or separate noise from the signal. And as far as processing is concerned, I'm not at all a statistician type person. In fact, I hate statistics. I mean, they're important, but I'm not the type of person who I leave that to the experts. I do try to develop interesting processing methods, though, and I sort of like to see interesting methods for processing, so that need statistics, but also are interesting ways of pulling out features of the data. So I'll talk in terms about that. I'll try to get to that today. So to begin, the whole talk will just be on contrast, resolution, paradigms in processing. And to begin with contrast, I covered a little bit of this in the introductory class, and just to let everyone know, there's much more to fMRI than just bold contrast. There's a lot of information that you can get from the fMRI signal that's related to functional contrast, starting with blood volume contrast with exogenous contrast agents, gadolinium, bold contrast perfusion. From looking at the ratio of bold to perfusion, you can get the change in metabolic rate. You can get the change in blood volume using vaso techniques. And this is a non-invasive approach that I'll talk briefly about. These are sort of experimental approaches that are still, I would argue, are still not really proven completely. Temperature actually has been somewhat proven, but it's really difficult to do properly. Neural currents theoretically has been shown, and it's been shown in phantoms, but never really convincingly shown in people. It's been shown also in cell cultures as well, but never in people. And diffusion coefficient imaging, people have claimed to see diffusion changes, but once again, it hasn't really been repeatable, and there's some debate as to whether it's from intercellular diffusion coefficient changes that occur with cell swelling or just a vascular effect. So these are sort of experimental, ongoing work. But to begin with just blood volume, just because I like to start with this because it illustrates basics of susceptibility, which I showed before. It also gives a historical perspective because this was actually, hold on a second, I'll just turn off my phone so no one calls me during my talk. This was actually the first successful results of fMRI et al. And once again, it involved an injection of a contrast agent that it took about 15 seconds or so to reach the brain. And then caused a signal decrease as it washed through. And then generally, if you keep it in there, if you watch long enough, it will change the T1 as well. Gadolinium is both a T1 and a T2 star contrast agent. But looking at the bolus as it washes through, this causes a massive dephasing in the signal. It's a susceptibility contrast agent. It's something like suddenly if your blood became deoxygenated, would occur. So during activation, you have a shortening of the arrival time, and you also have an increase in blood volume. So if you subtract this map from this map, you get something like this during visual stimulation. This is the back of the brain, and this is the cover of Science. It came out in November of 1991. And once again, just to illustrate this point, you can think of vessels containing susceptibility agents as having gradients set up around them. They distort the magnetic field. And if there's a gradient here, if you have a spin or a proton that's right here, it's processing faster than a proton right here. After a short amount of time, they'll dephase. And that's what causes this dephasing. So as the gadolinium rushes through, you have this steepening of these gradients. Then as it rushes out, you have a flattening of these gradients. And that's essentially what happens. So with bold contrast, just to reiterate the entire cascade of what happens, with tissue activation, you have local vasodilatation, causing an increase in cerebral blood flow and volume that causes oxygen delivery to exceed the metabolic need. The exact reason for this is not clear yet. You have an increase in capillary and venous oxygenation because deoxyhemoglobin is paramagnetic and oxyhemoglobin is diamagnetic like the rest of tissue. You have a decrease in susceptibility-related dephasing, which causes an increase in T2 and T2 star. And this leads to a signal increase in T2 and T2 star weighted sequences. And just to delve a little bit more into that, just very, very quickly, this is actually your typical T2 star decay curve. And happens to be at maybe a lower field strength than 3 Tesla. But if you have a T2 star of 50 milliseconds during activation, a T2 star of 48 milliseconds during rest, this is actually the inverse of the 1 over T2 star right here. Then you have a small difference in these exponentials. But it turns out exponentials are interesting. So it turns out the maximum difference, the difference in these exponentials is actually maximized at the TE equal to the T2 star. And so there's a shallow peak of this curve. And so when you choose an echo time of, let's say, at 3 Tesla of 30 milliseconds, that means that you assume that the T2 star, the resting state T2 star, is 30 milliseconds. But it's very forgiving as to what echo times you can choose. So you can choose out to 60 milliseconds. It should be fine. This is actually T equal 50 milliseconds right here. You can choose something out to 80 milliseconds and then as early as 10 milliseconds and still get a pretty good contrast. So there's a peak here, but it's a very, very shallow curve. More interestingly, if you look at the percent signal change versus echo time, it keeps on going up. And some people would argue, oh, well, let's just have a larger percent signal change. Let's go out to a TE of 100. Well, the contrast is then sacrificed at 100. And this is what really matters, is the contrast, not the percent signal change. But what's interesting, though, and this is actually something to consider, is that the T2 star varies depending on where you are in the brain. At the base of the brain, it's very short. You can have a T2 star of about 10 milliseconds at the base of the brain. In nice clean areas of tissue, motor cortex, it can be 60 milliseconds. And so this is actually looking at just a simple comparison between, let's say, 1.5 and 3 Tesla. And you can see that the optimal contrast is always, let's look at this top curve here. It's always optimized. This is with a longer baseline T2 star. There's a higher contrast. This is assuming a change in T2 star that's the same. If the baseline T2 star is longer and the optimal T2 star optimal TE is at the base, is at the optimal echo time, then you optimize the contrast. But it turns out that you can have much lower contrast than at 1.5 Tesla if you have a baseline T2 star, which is short. So you might be, in fact, if you're actually sitting at echo time of 60 milliseconds at 3 Tesla and your baseline T2 star is, let's say, 20, you'll have hardly any contrast left where you're looking. So that's a problem, though, if you're trying to look at base of the brain contrast and also motor cortex contrast. You have to somehow hedge your bets and maybe shoot for a slightly shorter echo time. So, okay, but at the optimal contrast, at the optimal echo time, 3 Tesla is always better than 1.5, and it's approximately double the contrast. It goes up about linearly with field strength. This is the baseline T2 star as it gets longer, and this is the baseline T2 star here. So, but one thing that is once again important, that if you have a very short T2 star, you're not really gaining any contrast at 3 Tesla. So it might be either better to shim or go down to 1.5 Tesla if you really want to get base of the brain contrast. So just a quick comparison of spin echo and gradient echo. If you have your whole pulse sequence right here, typically with echoplanar imaging, we just do the gradient echo. So we look at this first decay curve. But if you apply 180-degree pulse, you'll get your signal back. It's like a time reversal, and all the spins rephase, but not completely. There's some amount of dephasing that still occurs that's irreversible. That's your T2 dephasing. And if you compare contrast, in which you collect an image at the peak of this curve versus during this exponential decay, it turns out that the contrast is sensitive to different sizes of susceptibility compartments. So it turns out if the compartment is too small, then you'll have spins averaging out all the susceptibility. So they'll all still have the same phase. So there'll be no dephasing. This is for anything below 3 microns. But 3 to 15 microns is a sweet spot for spin echo sequences. And then anything larger than 15 microns, you have selectivity for gradient echo over spin echo right here. And just a quick sort of comparison of this contrast. This is with gadolinium, but still the same idea. We use a sequence that simultaneously collect gradient echo and spin echo data. And we just did a simple injection of bolus injection of gadolinium. And you can see with the gradient echo, you have selectivity towards these large vessels. It looks very clumpy. And with spin echo, you have much more of an even distribution of the contrast around gray matter right here. So just sort of illustrates the difference between gradient echo and spin echo. One other type of bold contrast that I'm not really talking about, but has been first was revealed by Ravi Menon, and that is that if you look at your anatomic images that are collected at a longer echo time, you have all these dark dots all over the place. And that's pretty much because the T2 star of blood, especially at high field strengths, is much shorter than gray matter. At 1.5 Tesla, it turns out the T2 star of blood is about the same as gray matter, so you don't see much. But at 3 Tesla or higher, you see these dark dots everywhere. And those are where the large vessels are. And you could actually make nice maps. This is a nice, one of the first maps made. Mark Hickey sort of pioneered this approach. He called it susceptibility-weighted imaging. It's a nice way of illustrating or revealing the venous vessels. This is a minimum intensity display projection through a slab. And this is some work from our group right here a while ago, also revealing the same thing. And these images get better and better as you go to higher and higher field strength. Okay, just to briefly talk about perfusion imaging. As I mentioned yesterday, there's many different flavors of how to do perfusion. There's many different ways of how you tag the blood flowing in versus how you normalize. These are just two. One is known as epistar, where you tag below versus tagging above. Fairs, where you tag everywhere versus just looking at the slice. And then if you subtract the pairs of images, this is where the blood is tagged flowing in versus where blood on this side is tagged. And presumably it doesn't flow in from this side as much. If you compare these images across the amount of time you wait between the tags, you get images that look, this is just highlighting the most rapidly flowing blood. This is only waiting 200 milliseconds, where you get, it's something like an angiogram. As you wait up to a second, you get more of the perfusing spins flowing in. So that's a really nice technique for looking at baseline perfusion. These are some of the first papers in this. Like I mentioned on Friday, it was pioneered by Koretsky's group, also Bob Edelman, Sanji Kim, who quantified FAIR, but Edelman coined Epistar. Ken had a technique as well, Ken Kwong. And just to look at the differences in terms of activation between perfusion imaging and BOLD. And this is kind of an old slide, but it still holds. Basically, this is your BOLD image, anatomic image. This is perfusion, and the contrast isn't the best on the slide, but this is during rest and during activation. You don't see too much. This took about 3 minutes to obtain. This took about 7 minutes to obtain. And it gives you kind of a sense of the comparison and contrast to noise. BOLD images look really nice. Perfusion are generally noisy, and if you blow it up though, you can see, even though the perfusion images are noisy, the hotspot of this perfusion image is very different than the hotspots of the BOLD at this resolution. So you can see a perfusion image shows a hotspot very much away from where the BOLD hotspot is, which seems to overlap if you look at the anatomic image where the dark dots are. So this is a vein, likely. This is also a vein. And perfusion is more where there's more of an even distribution of capillary changes. So it's great for localizing to capillaries. Not really critical for most studies that are just image at 3 mm resolution anyway, but pretty noisy as well. Except perfusion has a unique advantage of being really stable over time. So this is a nice paper by Aguirre, 2002 neuroimage, which he actually showed. So this is different on-off frequencies, and this is the contrast comparing perfusion to BOLD. And BOLD is at least twice, well, almost twice as good as perfusion. But it turns out that at very, very, very slow, this is going to faster and faster frequencies. This is going to very slow frequencies right here. At very slow frequencies, perfusion just stabilizes out, whereas BOLD gets completely awful because of the fact that in BOLD contrast, there's lots of low frequency drift. And it's hard to just regress that out because if you're looking for low frequency BOLD, you might regress out your BOLD signal. But in perfusion, it turns out that it's so stable, you can actually still create activation maps at very, very low frequencies. And this was a really nice example of comparing ASL and BOLD right here. And BOLD is actually, if you have 30 seconds on and off, 1 minute, 2 minutes, it still holds up. But at 4 minutes, you don't get much contrast. 8 minutes, nothing. Perfusion, you still get contrast, even 1 hour off and 1 hour on. And even 24 hours. So they weren't in the scanner for a day. It was basically put them in the scanner one day without doing anything, put them in the scanner the next day, have them tap their fingers, subtract those perfusion maps, and you get a really nice map of activation. So if you really want to look at slow, really nicely controlled comparisons between one day to the next and so on, you might want to use perfusion imaging. Okay, now looking at vaso very, very quickly. I already showed you on Friday the really high resolution vaso maps, but just to give you the basic concept, it was pioneered by Hanzheng Liu, who was at Johns Hopkins, and that was about 2003 in this paper right here in MRM. The idea is this, is that you do an inversion recovery pulse where you invert all the spins, and turns out that the T1 of blood is slightly different than the T1 of tissue. And it turns out if you invert the spins, as they recover, as the longitudinal magnetization recovers, it goes through a null point, and where there's no signal at all. And if you wait just the right amount of time, where the blood goes through, this is in red, it goes through the null point, turns out the T1 of gray matter, or the rest of tissue, is slightly faster, so that you'll have some signal left over. So if you then do an excitation at this point in time, you'll have no blood at all, and then you'll just be imaging tissue. The idea there then is that if you have an increase in blood volume, you'll have more of nothing, and the signal will go down. So you have more of a filling the voxel with a signal void from the blood, and the signal will go down with activation. So that's the idea. So this is bold, signal goes up, and vaso, during activation, the signal goes down. And that indicates the increase in blood volume independent of bold contrast. And so I and many other people often thought at this time that it was mostly just big vessels expanding, but it turns out that it probably is not that. Given the success at very, very high resolution applications of vaso, it's probably, the blood volume increase is probably due to recruitment of more capillaries with activation. And so that gives you a nice, highly focalized area of activation along the lines of perfusion, but might even be better. Okay, let's talk about resolution. Once again, I'd like to throw up this classic slide by Churchland. I think Patricia Churchland first created this. And with MRI, even though we fill this huge niche of temporal and spatial resolution, which is very useful, we're still always pushing this envelope right here. And not only in terms of the acquisition, not only in terms of how fast we can acquire our data, but also what is the true functional contrast, functional resolution. So the first thing, though, let's talk about the acquisition very briefly. With echoplanar imaging, which is pretty much established as being the most stable of techniques, it turns out that if our T2 star, let's say our T2 star decay wasn't 50 milliseconds, let's say it was 200 milliseconds. If it were 200 milliseconds, our resolution would be much, much better than it is now. I mean, there are strategies for going to higher resolution now using sensitivity of coils, which I'm not going to get into at the moment. But we're limited, though, by how fast this signal decays. And so we have to collect our data as rapidly. First, we have to wait until the optimal time, and then we have to collect as rapidly as possible before the signal decays. If this signal took a little bit longer to decay, we could spend all this time acquiring data, and that would allow us to make a nice single-shot echoplanar image. But it's unfortunately, in this case, nature kind of works against us. And so we're constantly struggling to collect data as rapidly as possible before the signal dies away after about 100 milliseconds. And okay, so that limits us in terms of our spatial resolution for echoplanar imaging. What limits us in terms of our functional contrasts, both spatial and temporal resolution, is the hemodynamics. And it's more complicated than just saying the hemodynamics. It's simply because of the fact that hemodynamics vary. So this happens to be like, let's say, a set of voxels over this small area of cortex right here. You might have some areas that are purely arteries, some that are capillaries, which is about maybe 2% blood volume or 5% blood volume, some that are pure veins. We could have some voxels that are 100% veins. So this affects your signal magnitude, this affects your signal latency, as I'll get into. And then, of course, the noise affects it as well. But, of course, the highest functional resolution that has been obtained has been on the order of ocular dominance columns, which is about 1 millimeter or 0.5 millimeters by Ravi Menon again and Kang Cheng. And this is, as I mentioned on Friday, really nice work by Essa Yacoub in their paper in PNAS, in which they were looking at orientation columns. So these are ocular dominance columns, and then within the ocular dominance columns, this is basically a center point of a pinwheel of orientation columns as basically a stimulus, a graded stimulus was rotating, and this is color-coded according to its phase of rotation. And that's just a simple map showing the raw activation as the stimulus rotated. Okay, so that's kind of represents the highest spatial resolution. Of course, you have layer effects that I showed as well, which is sort of at the same spatial scale. Okay, now let's go to temporal resolution. Before we go to temporal resolution, so once again, this is, and the reason why these were obtained, this is 7 Tesla using spin echo techniques, and the layer-dependent activity was using vaso techniques, sensitive to capillaries at 7 Tesla. So that's kind of key to getting to this resolution. And one thing actually I just want to re-emphasize with this resolution is that this might be the highest resolution at which even the hemodynamics are controlled by neural activity anyway. So even if we can go to higher resolution imaging, even optical imaging data, which is much higher resolution, still, this is pretty much the highest functional resolution that optical imaging typically describes anyway. So we might be at this might be a great thing, that the upper limits of sensitivity of fMRI match the upper limits of hemodynamic control in the brain. Okay, so let's go to resolution, temporal resolution. So starting out with temporal resolution, let's look at the very slow end. Turns out that there was some controversy in the early days as to if the bold signal actually stayed elevated with sustained activation. And it turns out some people thought it didn't. I think the conclusion finally was that it actually did. As long as neural activity was going on, bold signal was maintained. And this is an early study that I did in which we first did, control 1 minute flashing checkerboard. And then I put people in the scanner and had them stare at a flashing checkerboard for 25 minutes, which actually some of them never volunteered ever again for a study after that. But we got our data, and it turns out that we also collected flow data with this as well. And it turns out the flow and the bold, as long as flow stayed up, bold stayed up. So it's nice to know that there's no sort of habituation, at least on this time scale of the hemodynamics. Let's see how short we can go, though. So this is just some data that I got from collaborating with Ted Dio a long time ago, in which a stimulus down to 250 milliseconds was given. We have even gone down to 16 millisecond stimulation and still obtained a response. So there's really no limit to how brief of a response you can give. It's more limited by, 16 milliseconds we chose because that was a refresh rate of the projector. So it'd be interesting to see how brief you can actually go and get a response. I'm sure there's ways of getting more precise timing, but then it gets to the point where you can't even see it. And I don't know whether or not that would be relevant. Another way of thinking of temporal resolution is how fast you can turn on and off the signal and still elicit a response. So these are some old data in which we went from 24 seconds on and off down to about, at 6 seconds on and off, it turns into like a sine wave. At 3 seconds on and off, it becomes more saturated in the on state because it takes a little bit longer to return to baseline. And it doesn't really keep up past 1 second on and off. And this sort of shows the filter properties. Basically, this is the on-off cycle length. And it sort of has this elbow at about 16 seconds, 8 seconds on, 8 seconds off, before it starts reducing contrast pretty rapidly. However, there was a recent study by Jonathan Polemini in which he had pushed it even further and showed that at 0.5 hertz, if you go on and off fast, on and off at 0.5 hertz, you still, if you look very closely and do a lot of averaging, you can still elicit this response. And he recently just even pushed it to 0.75 hertz and still elicit a response. And I mean, this is interesting because I think there's something interesting to look into here, that the signal, and he was basically describing that it's what they see is not expected because it seems to suggest that the bold signal is very nonlinear. And at very rapid on-off signal changing rates or on-off neural activation, you still see a signal. And we also know that it's very nonlinear because at very brief stimuli, you get a larger than expected event-related response. So they could be related in some regard. And I'll get into that in a couple more slides later. But another way of thinking of the hemodynamic response, the temporal resolution, is in the way that we all like to think of it, is if one part of the brain turns on, if two parts of the brain turn on with a slight delay, can you resolve them? And it turns out that's a lot harder because of this. So this is, once again, a very simple experiment in which we mapped, basically it was finger tapping for 1 second, and we elicited a nice hemodynamic response. We did a very crude way of looking at the latency. We shifted this response function and then mapped out the voxels, mapped out the time delay at which this was the highest correlation between this function and the actual hemodynamic response on a voxel-wise basis. So we actually were able to map the latencies that way. Turns out if you do it that way, you get a range of latencies between plus or minus 2 seconds. And here's the distribution right here. So that's a large distribution of latencies, even in looking at an area as small as the motor cortex. And it turns out the areas that show the longest latencies do appear to be right here, where there's this large vessel right here. That's the longest latency. And then if you look at the magnitude, it seems to also show the largest magnitude. So it seems to suggest that these are large vessels that have this long latency. So maybe one way of reducing the width of this distribution is by getting rid of the large vessels. That's one way. Other ways might be more, getting capillary selective techniques. Another approach, though, is to try to modulate the timing of the experiment. And not to get absolute latencies, but get relative latencies with the timing modulation. So here's an experiment, once again, kind of an old one, where we did a hemifield experiment where we turned on one hemifield 500 milliseconds before the next, and then turned it off 500 milliseconds before the next. And if we pick our regions of interest just right in the visual cortex, the left and right hemifield, we see this onset difference and this return to baseline difference. And one thing actually I just want to emphasize as well that people don't usually comment on is that the error bars during the onset are smaller than during the return to baseline. So there's much less variability in the hemodynamic response when you're driving it than when it's sort of relaxing down to baseline. And then, but the problem is with that, it all depends on how you pick your region of interest. And so you don't like to, I'd rather make maps. It's much more honest to have like these maps. So here's a map of the latencies with with the 500 millisecond delay right here. And it doesn't really look like much. It just looks, because it's dominated by the intrinsic variability in the latencies. And so what we decided to do is switch the order. So this is the 500 millisecond latency in which we switch the order of the hemifields. So now turning on the right and then the left. Also, this map doesn't look like much. But if you subtract the maps, you get a really nice differentiation between left and right hemifield. And so what that indicates is that even though there's this massive spread over space and the latencies, the hemodynamic response is well controlled within each voxel. So if you modulate it a little bit, any change in latency that occurs is reflected in the hemodynamics. And we've even gone down to 250 milliseconds, and we see a pretty good response. So, and once again, Ravi Menendez has went down to 50 milliseconds and able to pull that out. And we tried to apply this with one paper that we were able to pull off. This is with Pat Belgan when he was a postdoc of mine. And the idea was this, is that we tried to model the hemodynamic function in terms of, okay, so if you have an increase in the width with a task modulation, if you have an increase in the width, that seems to indicate that there's upstream sort of effects that cause it to be slowed down. But the delay is not, there's no delay. But if there's an increase in delay, then then it affects the latency downstream. So you can either have the increase in delay or increase in width. And the basic task was this. It was a word recognition task and rotation task. So it turns out that your reaction time changes if you, as a direct function of the angle at which words are rotated. And so that's very well described here. So this is 800 milliseconds up to 1400 milliseconds. And it turns out the reaction time also is delayed depending on whether the word is a real word or a non-word. It takes a little bit longer to recognize it as a non-word. And so the idea then is that we had these two tasks going on, and it turns out that when you compare the word versus non-word, and this is the left prefrontal area that's presumably a word generation area, turns out the word versus non-word, with the non-word, it takes a little bit longer. It has a slightly wider width of this curve. So that means that this might be the bottleneck that's doing more processing to generate, to decide whether it's a word or non-word. But it turns out with the rotation, there is no width difference, but it's actually, I'm sorry for the poor quality of this, but there's no width difference, but it turns out there's an onset difference. So if you look at the rotations, it seems to suggest that this is downstream from some other area that's doing the rotation that then feeds it into this area that then does the processing. But the width doesn't change. So this might be a way of sort of pulling out where certain areas of the brain do what, depending on the width or the onset difference with timing modulation. Okay, so let's say in an ideal world, there is no hemodynamic delay. There's no delays. Everything's perfect. You're just dealing with signal to noise. And you have a certain signal change. This is a nice simulation that was done by Rasmus Byrne a while ago. 1% noise, 4% bold, 256 time points per run, and 1 second TR. What would be the upper temporal resolution if you had no hemodynamic effects to deal with? It turns out that if you plot the smallest latency variation detectable with a p-value down to 0.001 versus number of runs. So if you have also the timing, it turns out that if you had 8 seconds on and 8 seconds off, that seems optimal for detecting latency differences. And you can get down to about 100 millisecond onset difference with about 11 runs, which is about the amount of time of a typical scan session. And turns out that it would take about 20 with a 16-second on, 16-second off. So, and you can get down to about 50 milliseconds if you average for about 30 runs. So that's sort of like the upper limit of the temporal resolution as we know it, as far as the signal to noise limited. So there's one other approach that I've always thought was very clever, and I still think is still relatively unexplored, and that was provided by Seiji Ogawa in a paper at PNAS, in which he looked at the magnitude of the somatosensory response in rat whisker barrels, or I'm sorry, rat forepaw, the areas corresponding to rat forepaw stimulation, and looked at the relative magnitudes. So it turns out if you stimulate one forepaw, that will suppress the activation from the other forepaw at a very specific timing. So it turns out that if you wait too long between the timing, there's no effect. But it turns out that if you get the maximum attenuation, if you stimulate one forepaw and then wait 50 milliseconds and stimulate the other forepaw, you'll get the maximum attenuation of the other forepaw signal by the first forepaw input. So that's sort of, and basically he's mapping this attenuation curve with the relative forepaw timing. And he's able to map about a 50 millisecond timing delay between left and right forepaw. So basically, even using the slow hemodynamic response, you can map very, very rapid communication, inhibitory communication. So as long as it's inhibitory, or it could be excitatory, as long as it modulates, you can map maybe very rapid timings by adjusting the timings and looking at the amplitudes as they modulate. So I thought that's an interesting direction to go. Okay, so now I'm just going to combine paradigms and processing in this last 20 minutes. See how far I can go with that. And if I run out of time, I'll still just stop, but I'll just mention that the slides and everything are online, so you can just download them. Okay, so over time, a bunch of different types of paradigms have evolved. Block design, more esoteric things like frequency and phase encoding, event-related design, fMRI adaptation, orthogonal block design, which is a little bit more esoteric, and some of these are very much related, and then free behavior design. Just to quickly go over block design is the most basic type of stimulus, and it's one of the first ones that was employed, sort of based on PET imaging timing, in which you give sort of a 20-second off, 20-second on sort of timing. And you can look at, this is a really old paper, but it sort of illustrates the point that it's very forgiving in terms of how you choose a reference function. You can pick a middle image, you can pick a boxcar that's shifted in time, you can pick some sort of smoothed reference function, or you can look at the data itself, and you get maps that look all kind of similar. Or else you can take the Fourier transform of the data. In this case, this is like maybe fitting it to a sine wave, which is taking the Fourier transform and looking at the power, which is right here. So it all is pretty forgiving, at least in motor cortex. It looks more or less the same. Some of these you might emphasize some noise or not. Like here, you fit to the actual, looking at A, you fit to the actual noise of the function, or I'm sorry, looking at B. But anyway, that's sort of looking at various reference functions, very forgiving as far as that's concerned. But one thing I want to emphasize with the block design is that most people, if, for instance, in this particular paradigm, it was left finger tapping for 20 seconds, right finger tapping for 20 seconds, and so on. But I think actually a lot is missed when you choose your reference function or you choose your regressor. And I think this is a nice paper that illustrates that point, is that, you know, a very simple stimulus, so it's right checkerboard stimulation, right hemifield checkerboard stimulation. And the blue function is what you'd expect to see in visual activation. And you get, of course, as you expect, a left visual cortex activation right here. But it turns out if you just pick a reference function that just covers the two transients, the on and the off transient, you get a nice bilateral activation more in the periphery and also in the insula, or insular sort of area here. And that's also activation. It just doesn't happen to exactly match the timing that you necessarily model. So it's always important to try to, if you're designing models for pulling out your data, to sort of throw in other models just to see if you get a systematic effects. In particular, the transients at the beginning and the end are important. And this is a paper by Javier Gonzalez-Castillo that shows that, in fact, there are all kinds of real activations that have different shapes. This is, for instance, this is averaged over 9 hours, the same subject across multiple days, and you see, you know, this is a very simple visual task, visual motor task, and you get, as you'd expect in the motor cortex activation here, in the visual cortex, you get what you would expect to be the activation right here. But everywhere else, you get all kinds of things. You get negatives, you get positives when the signal turns off, you get transients. There's all kinds of different types of activation that are still there in a boxcar or a block design. Okay, for frequency encoding, this is, I think I'm the only person who sort of pursued this just because I thought it was a cool thing to try. But it's kind of fun, and it might lend itself to certain types of stimuli. In this particular case, I had a subject in the scanner and verbally cued them to tap their fingers at one on-off rate, and then tap their other fingers at another on-off rate. Then taking the Fourier transform of the two time series, we have two separate peaks. And then using basically all I did, we could have just correlated with the sine waves, but we just decided to do it this way because it was insensitive to phase. We're able to pull out left and right motor cortex. So two different on-off frequencies, just pull them out after the fact, just frequency encoding. Phase encoding is much more common, and that lends itself to stimuli that can be continuously changed, that have a mapping to cortical surface that changes also continuously. For instance, retinotopy. So here's an example from Ted Dio's group a long time ago, in which he had a stimulus. This was a checkerboard that would expand, and he encoded the activation according to this colors right here. Whatever activation occurred with whatever ring expansion size that occurred is color-coded. And you can see that with an expanding ring, you can actually phase encode the data as the stimulus expands. Here's 3 different subjects. And you can also phase encode the data as the stimulus is rotated. And it turns out from here, from the rotation stimulus, it turns out that in V1, the direction of the activity goes one direction. In V2, it goes the other direction. So using this phase encoding as you rotate a stimulus, it turns out that you can map out where the borders between V1 and V2, V2 and V3 are, because the propagation of the activation goes opposite directions, and so they meet at the borders. So in this way, he was able to map out borders of visual cortex between V1, V2, and V3. Okay, event-related design. First, event-related design. Nobody really even thought you could do event-related design. Then Greg McCarthy at Yale, and then also Randy Buckner with this simple task of doing a word stem completion. So with each word stem, you had to come up with a word to complete it. He took a region of interest also from this left prefrontal cortex area and was able to show this event-related response with word stem completion. And it turns out that, so we were able to get robust responses. And for a short period of time, people thought, including myself, that, not thinking that the system is necessarily linear, that you would have to wait for 15 seconds before giving another stimulus to allow the hemodynamic response to settle down. Turns out we did this, and we found one thing. One is that if you wait a certain amount of time, the relative contrast relative to block design is about 60% of block design, which is much higher than you would expect also from a linear system. Okay. And also the optimal timing is about 12 seconds or so. And these are what the maps look like. It's just, they get worse and worse as you go wait shorter and shorter periods, of course. But the real revolution came with Anders Dale and Randy Buckner, in which they demonstrated quite nicely that, in fact, the hemodynamic response is linear and within this time frame. And so you can superimpose stimuli and then deconvolve them. So if you give one single stimulus, here's their paradigm. One stimulus, basically two that are separated by, this is 0 seconds separated by a second, and then three that are all combined all at once. And what they did was basically took the first one and subtracted it from the second one to get this almost the same hemodynamic response elicited from the second stimulus. And then they used the second one, subtracted it from the third one to get back also the original hemodynamic response. It's very fundamental deconvolution showing that the response is preserved even if the stimuli timing caused overlapping hemodynamic responses, which was great because it allowed you to go really fast. And so the optimal timing for event-related responses, it turns out is complicated, but it turns out that if you plot the detectability of the response versus the average interstimulus interval for various stimulus durations, this is a stimulus duration of 250 milliseconds. The average ISI is about 10 seconds. If you stimulus duration of 100 milliseconds or 1,000 milliseconds, 1 second, the average ISI is about 12 seconds. This is average, so you can vary from 1 second down up to 15 seconds, and so on. And it turns out, of course, as the stimulus duration gets longer, so a stimulus duration of 4 seconds. In this particular case, the average ISI is about I'm sorry, I messed this up here. So this is the constant ISI, these solid lines. Where you're using a fixed, assuming you're not deconvolving, and this cloud of data are simulations of jittered ISI. So it turns out, with a very short stimulus duration, you have the average ISI is like 1 second. With a 1 second stimulus duration, the average ISI is about 3 seconds. The optimal ISI and 4 seconds. It gets a little bit longer. It starts to approach block design as you go longer and longer. So that's for detectability. So it turns out it is generally better to have a longer stimulus and a little bit longer average ISI in terms of optimizing for detectability, which means making maps from your event-related data. But let's say you know where you're looking. If you know exactly where you're looking, if you pick a region of interest and you say, I really just want to characterize the shape of that response, it's a very different optimization, it turns out. And it turns out that the average ISI, so here, it's generally better to go longer, to stimulate a little bit longer and have a little bit longer time in between, although you can do pretty well with a 1 second stimulus. Here, if you're trying to estimate the hemodynamic response, you know where you're looking, and you're just looking at how that signal changes, it's always better to go shorter. So if you have a stimulus duration of 250 milliseconds, you get much better contrast for estimating the shape of the hemodynamic response if you go really, really short. down to 1 second average ISI. Okay, other advantages of event-related. You can completely decouple your motion from the signal. This is some nice work also by Rasmus Bern, in which he demonstrated with comparing a block design to an event-related design that involved talking, that the motion effect can be completely separated from the hemodynamic response, because the hemodynamic response happens on a much different time scale. So if you just speak, this happens to be a block trial in which the subject is counting, is saying words. And in that case, the motor cortex, mouth motor cortex is activated, but also you get all these edges all over the place. But it turns out that if you speak in an event-related fashion, you say a word, you wait, say a word, wait, you get a very, very clean, just the motor cortex response. You don't get the edges, because the edge response is completely decoupled from the motion response here is completely decoupled from the hemodynamic response. Okay, and this is a graph that summarizes all the timings between event-related and block design that optimize contrast and minimize to noise. So it turns out that the further you are away from this 0 point, the more you're sensitive to motion. And along here, the more you're going into the direction on the right, the more you're sensitive to bold. So, and this is basically various types of block design is in brown, stimulus duration of 2 seconds, 4 seconds, 6 seconds, 8 seconds. A block design at 10 seconds is really optimal for separating from motion. It's almost perfectly orthogonal to a motion function. And it turns out that for event-related, you have you'll get a little bit more sensitivity and stay around 0 for event-related for varying ISI and so on. You can study this graph later. I'm not going to go into details just for the sake of time, but it's a nice laying out of differentiation from artifact. And here's just the maps to show it as well. Okay, fMRI adaptation. Okay, so it turns out that this is a nice paradigm that was actually pioneered by Kalanick, Grills, Spector. This shows the idea, and that is if you give a stimulus, it takes a certain amount of time for the neurons to recover. And if you give that same stimulus a little bit later, while the neurons are somewhat habituated, you'll have less of a neural activity from that neural pool. So the idea is that if you look at the amount of signal change between the first stimulus and the next stimulus, and if there's no signal change, that means you've given a different enough stimulus such that you're activating a fresh pool of neurons. Whereas if you get this habituation here, you're activating the same pool of neurons as before. And so this is a nice way of pulling out within a voxel what pools of neurons are selective to certain stimuli. And here's one example in which Kalanick, Grills, Spector's group, first on the first one did all cats, for instance. And if you do all cats, you get something like this. And if you do some categories that show different pictures, but I mean, if you interleave slightly different images, depending on the neural responsiveness, if it responds in the same way to the image or not, you'll get different amounts of habituation. So here, you'll have a little bit less habituation. If you have even more different pictures, you'll have less habituation. And then finally, if you have still almost completely different pictures, you'll have less habituation. So I'm not going to get into the interpretation of this other than they're trying to pull out how the brain responds, how basically it groups and responds to categories. So it's a really nice way of doing that type of differentiation. But I would even argue, though, that the brain habituates all the time for everything. So just as an example, as I mentioned before, the nonlinear hemodynamic response with a very simple visual stimulus shows a much larger than expected response at 0.25 seconds, but behaves in a linear fashion at 20 seconds. So it's very nonlinear. It shows basically, if you tried to look at the nonlinearity, the hemodynamic response with the visual stimulation, it's extremely nonlinear up to about 2 seconds. So there's still, this is habituation as well that's going on in the visual cortex. You might vary your visual stimuli at this time scale to maybe pull out more low-level features, potentially. Okay, so looking at habituation effects is also an extremely effective way of pulling out differential neural activation. Orthogonal block design, it's a little bit esoteric, but I really liked it because there was a beautiful paper by Susan Courtney in which she she actually made it, she was looking at working memory, and she was trying to come up with 6 orthogonal functions that separated, and it's hard to read, but basically looked at non-selective visual activation, faces and houses, versus stimuli, let's see, face stimuli versus house stimuli, and so on and so forth. And you actually get very specific types of contrasts all in one time series. It's always much more powerful to pack as many different types of contrasts into a single time series than to spread it out among time series, because there's a certain amount of instability that comes across from one time series to the next. So if you can pack as much in as possible, you're better off. And so here she packed in 6 different comparisons, or let's see, 1, 2, 3, 4, 5, 6, 8 different comparisons, and was able to, because these functions are all orthogonal to each other, it means they're mathematically, their correlation between them is 0. So you can pull out very cleanly the various maps that correspond to the functions that correspond to those specific comparisons really nicely. So anyway, it's a nice paper just to take a look at in terms of designing paradigms for orthogonality. And there are ways of actually making any 2 time series orthogonal mathematically, but you give up signal to noise if you do that. It's much better to try to make them orthogonal from the start as opposed to orthogonalizing them. So, okay, lastly, free behavior design. Here it sort of represents the natural evolution of I think, fMRI designs where you're getting more and more complicated. You're allowing the subject to behave more freely in the scanner, but you're keeping track more precisely of what they're doing. And so Eleanor McGuire's group was one of the pioneers on this. And so here they had a simulation in which they had somebody driving through London right here, and here's the map of where they're driving through London. And then they tried to categorize and bin various types of categories of decisions of what they were either experiencing or what they did. And there's basically the bins right here. And if you bin that data and then compare it to all the rest, you actually get something that looks like this. You get an activation map corresponding to whatever category of what they were doing freely. So they let them freely do whatever they want and then just keep track of whatever they did, bin that data together, and then compare, then make maps from that. So it's a nice way of sort of efficiently allowing the subject to freely behave as long as you keep track. Another one of the more beautiful studies that I've seen is a monkey study with logothetis, and I still would call it free behavior even though the monkey was sleeping. But here in this case, this is sort of illustrates also the use of multimodal integration. This is basically using electrophysiologic measures of the hippocampus during sleep. And all they were doing was averaging the data whenever ripple activity occurred in the hippocampus. And if that occurred, basically they would map out, they would convolve with a reference function and map out the area of the brain that corresponded to the ripple hippocampus activity. And he found very clear activation, that cortical activation that always occurred with hippocampal activity, and subcortical deactivation that always occurred with hippocampal activity. So very nice study. Once again, freely behaving. Real-time fMRI. So I'm getting towards the end. The idea here is that this is a nice technique that allows the subject to receive back feedback based on their brain function. So brain function happens in real time. You have an analysis that's fed back to the subjects, and they're allowed to do something with that stimulus. Okay, and so it turns out subjects can do things with various, if the right type of hemodynamic feedback. In this case, this is some work by Rainer Goebel's group, in which he looked at depressed patients and found the network of activity, the network that was associated with emotional arousal, and asked the subjects to try to increase, based on feedback, that emotional arousal network. And he found that after a few weeks, it actually worked pretty well. So this is before and then after the therapy, and the red is after. And their depression score consistently decreased after therapy by learning to increase their emotional arousal network. And it's actually in a clinical trial in the UK. And this whole field sort of got off the ground. I think that with Christy Charm's paper, in which he actually helped people who had chronic pain in their back. So chronic pain, first he mapped out the areas associated with pain, here and here, and asked the subjects to decrease the activity in these specific areas by simply having an illustration of a fire that was either lower or higher with the activity. And so they were asked to try to decrease the fire. They figured out ways of doing this, and it turns out that the amount of success that they had in decreasing this activity corresponded to their internal perception of their pain. So, and it actually lasted for a few months after that too. So it's potentially a nice way of figuring out, not just simply based on biofeedback signals from peripheral nervous system sort of activity, but actual cortical activity, and allowing yourself to somehow work out a strategy for modulating that. Takes a while, but you can actually do it, and it seems to have effects. Okay, wow, I'm not going to cover this last two parts. Many people will be talking about resting state activity over the course of this whole course. So I'm not even going to, there's enough people talking at it. I have the slides here if you want them. fMRI decoding, we have several lectures on decoding and actually encoding and even more advanced work than this. So I'm not really going to touch on that today either, but at least they're here in the slides. And just to suffice it to say that resting state fMRI is sort of even a further extension of free behaving fMRI, where you're just allowing the subject to either stare at a fixation and then figuring out ways of pulling out correlation in the data. It's really taken over most of fMRI, or at least half of all of fMRI now is looking at resting state correlations. So it's extremely powerful, it works very well, and we're still trying to figure out how to deal with the data. fMRI decoding and encoding is nice in the sense that it doesn't really, it lends itself to studying the brain where just simple mapping fails to capture the essence of the information there. So we're used to looking at these blobby maps where you have motor cortex and visual cortex activation, but if the pattern becomes so fine and distributed and variable that it just looks like salt and pepper to our eyes, it doesn't mean much to us. But if we use it if we look at the correlation between that salt and pepper pattern with another activity, we can say, oh, well, there's actual different brain activity there, even though it doesn't obviously appear this way to our eye. So fMRI decoding allows you to do that, to look at very fine-grained multivariate responses that our brains just simply don't say, oh, there's activation. So anyway, I think I'll just leave it at that. Yeah, and if there's any questions. Any questions? Oh. Okay. Yeah.
I have a question. So regarding the stuff that you showed about, like you showed checkerboard, and on one side of which you feel, and then you could do a block design, and then you do like more of the whole block, or just the beginning and the end.
Yeah.
How much degrees of freedom do you have there? Because I'm wondering whether you actually capture different psychological processes.
Wait, how much of what?
How much degrees of freedoms do you have in like picking your functions? Because you might actually model completely different psychological processes.
So when you just turn on a checkerboard?
You show a checkerboard, and then you show like when you do like when you model like the response of the whole block.
Yeah.
Because it's the onset and the offset.
Right.
how much degrees of freedom do you have in choosing your functions, because you might model very different psychological processes. So, for example, something like visual processes versus attention on processes.
Yeah. That's sort of an open question. I mean, you can think of degrees of freedom in terms of how correlated the fMRI data are, and that's not even really accounted for. But degrees of freedom in terms of timing of psychological processes, I don't even think most people even think about that in terms of. What's that?
It feels really important in terms of what you want to capture.
Oh yeah. No, actually, I think that in this particular case, we just use either boxcar, which assumes it's just low-level visual processing, visual activation, or the transients, which might be the more psychological, might be more of like, a set point of like, let's be aware of this stimulus. Right, and there might be some things in between as well, and that may be worth studying. I just wanted to bring out here that it's always better to keep an open mind in terms of putting in your reference functions. All right. Okay. Any more questions? All right. Thanks. And the next lecture will be on Wednesday. So, all right.